People often ask me what it was like to work during the time of the Human Genome Project. My role at the DOE Joint Genome Institute was the production sequencing department head. I started there roughly in 1999, and I managed the operations until 2013. During that time period, there was a big race going on between the public and the private sector. We had a private company challenge the public sector. The private sector thought that they could do it faster, cheaper than the public. There was a legitimate concern at the time that if the private company won the race, that the data would not be readily available in the public domain. And so the public had this opportunity to be able to show what they were capable of doing. So you used to walk into a room of just a sea of sequencers, these large machines. There was probably 100 or so in a room, covered the floor. And in each one of these sequencers was this set of capillary arrays that I'm holding here in my hand. And these arrays are what we used to decipher the individual bases of the human DNA. So how we were able to determine A's, C's, G's, and T's. Staff would have to manually load these instruments, so there was no major automation at the time. We would manually load the instruments from morning to evening, all through the weekends. One of the things that I really valued about working at the JGI was all the creative minds. And so when we had to tackle the scale of sequencing the human genome in a short period of time, the staff got creative and came up with ways to automate their areas. And so what I'm holding here is a pipette, which is a manual instrument used to transfer liquid from one receptacle to the next. The staff came up with a way to automate that and then submitted that automation into a competition that they then took home an award called the Ergo Cup. I want to congratulate JGI on their 25th anniversary and say how proud I am that they are part of the Berkeley Lab user facility ecosystem, supporting scientists around the world.